Noswaith dda, a chroeso I chi gyd. Well, good evening and welcome everybody to our session today. If you don't know me, I'm Professor Colin Reardon, Vice Chancellor and President of Cardiff University, and I'll be hosting the event tonight. As you've just heard, it is being recorded, and we'll send you the recording link after the event, and you're very welcome to share that if you find it interesting. So this evening, you're going to hear from our researchers on a topic that really concerns all of us, of course, how the world responded to the coronavirus pandemic and how our researchers responded to the pandemic. Now, you don't need me to tell you what happened. It's the one universal wherever you go in the world, wherever you meet new people. You can just talk about before and after COVID or before and after the pandemic. Everybody knows what that meant. The really surprising thing is that it seems so surprising. I guess it's because something hasn't happened within living memory. It's really too difficult to imagine it ever really happening. And so even though it was very well known that at some point there was likely to be a respiratory virus pandemic, somehow it took us all by surprise, as you well know. And it did, of course, require a global response with the global infrastructure we now have, with rapid transport and flights and so on. It did take a global effort to develop vaccines, which happened with really unbelievable speed, it has to be said, to work out and implement controlling tracking methods, and really for everybody to be educated as far as possible about how to handle this in terms of our own personal behaviour and our own daily activities, which, as you know, were extremely curtailed for long periods, and it was a pretty difficult time. And of course, the effects of COVID are still with us. It's not over yet. If you look back in history, the flu pandemic that took place or suspected to have taken place in the 1890s, and the one that definitely took place in 1918, it took around a decade for the echoes of that and resurgent waves to wash through. And of course, in those days, it didn't have any vaccines, and treatments were pretty rudimentary, treating symptoms really. But of course, what COVID has done also is to trigger many cases of long COVID, which is very distressing for those affected. And of course, there are other associated long-term physical and mental health effects. There are effects still on the health service here, in terms of the strains that the service was put under and missed treatments and so on because of COVID. So, we all lived through it, so we've all got our own perspective of what happened, but the story behind the scenes, I think, is something that's only really starting to emerge now, and it'll probably be quite some years before we really have a reliable history of the pandemic. But clearly, this was a devastating virus that had a huge effect on communities. It destroyed lives, and it took a huge emotional toll, which is extraordinary for something that's so small. I mean, viruses are really, really tiny compared with bacteria. You could get all the viral particles of COVID that are in the world now probably into a standard fridge, and it has this enormous effect on people. And of course, the economic consequences will be echoing through. They echo through now, and that will continue for years. And it's really transformed the life of cities and the world in other ways. Now, of course, without the global scientific response, the effects would have been much worse. So we are very proud that our researchers here at Cardiff University were able to play their part in the effort to combat COVID. And you'll hear about that shortly. Our speakers this evening are Professor Ian Humphreys, co-director and lead of infections and pandemics in the Systems Immunity Research Institute at Cardiff University, and Professor Richard Stanton, who's the head of infection in the School of Medicine at the university. Both of them contributed to the response, and their current research is going to help us learn from COVID to prepare better for emergent infections of any origin. And of course, now we've had one pandemic, we're aware that there could be another one at some point. Before we move on to their presentation, you may or may not know that This is my last year as Vice Chancellor at Cardiff. I've been here for almost 11 years, but I retire at the end of August, and on the 1st of September, Professor Wendy Lana will join us as my successor and as the university's first female Vice Chancellor in our 140-year history. Wendy's an excellent appointment. She's an internationally respected social scientist, and I'm sure that she'll be continuing to take Cardiff University along its very positive trajectory that we are now on. It has been a huge honour and a privilege to represent such a dedicated community of not just researchers and academics, but also alumni, students, staff, donors and friends who all support the university in various ways. The COVID pandemic, I think, really revealed the role the university can play and how much we need the support of you and the community around us. So it now gives me great pleasure to hand over to our speakers. So I'll disappear for a moment, and it's Ian and Richard. Welcome.
Thank you, and welcome everyone. Let me just get slides up. Okay, so welcome, good evening everyone. My name's Ian.
And I'm Rich.
And really tonight, what we'd like to do is really give you a flavour of some of the scientific research that scientists in Cardiff University have been doing during the COVID pandemic, and also give an insight into what we're thinking in terms of future pandemics and what we can do best to help protect humanity from these events. So when COVID really came to the fore in early 2020, in the UK and elsewhere in the world, scientists really launched into action on a number of fronts. So we were involved in helping advise Welsh Government in terms of various policies around COVID-19. We were involved in helping test and track the COVID, the coronavirus that causes COVID in the UK COG consortium. In Cardiff, we established our own testing pipeline for asymptomatic testing of students and staff. We worked a lot with NHS colleagues. So, for example, we used our high containment facilities where we could work with the coronavirus to help our colleagues in the NHS detect COVID in patients, for example, in ICU patients, to understand when the patients have actually cleared the virus and when they were safe to work with outside their high containment areas. We're also involved in disseminating information to the public. Certainly, there'd be times during the COVID pandemic that it'd be hard to turn on the TV and not see my colleague Rich telling the world about the latest scientific developments around COVID-19. But of course, we're scientists and we were performing a lot of research at this time to understand COVID as best we could. And certainly there are a number of us who are particularly interested in understanding the role the immune response plays during COVID-19. And that was important because it helped predict which patients would go on to have severe disease and which would do better. It helped to design therapies for the treatment of COVID. It also helped in terms of diagnostic development and also informing vaccines that ultimately have enabled us to move on and deal with COVID better in our everyday lives. Now, we certainly did this on two levels. We were doing our own research, but we're also leading national efforts in scientific research. So, for example, Cardiff played a leading role in the UK Coronavirus Immunology Consortium, which, as you can see from this map, really brought together scientists around the country to tackle this disease. We're also involved in the national core studies, which look more broadly at how COVID-19 causes disease in people and how we can help counter this virus.
So we thought we'd just give you a sort of bit of an overview of some of the types of studies we got up to in Cardiff. So this is a study here on your screens from one of our research teams here, who really asked the question, why is it when we've all been vaccinated against SARS-CoV-2 that some people still get infected while others are protected? And so we all know about antibodies, but this group here were interested in other parts of the immune system, in particular the T-cell response. And they were able to take blood from people and then wait and see who got infected and who was protected. They could then go back, look at the blood and say, what's different about the blood from the people that didn't get infected compared to those that did? And so in the graph on the right, you can see one of the major outcomes from their study, which was to look at this particular type of T cell and show that it was much, much higher in those that remained negative for the virus compared to those that eventually caught it. And that's really, really important because it tells us that if we can induce these type of T cells with our vaccines, they're going to be a lot more effective. Another thing we were very interested in is obviously we have immune systems in all of us that are generally effective. They have many, many parts which work very efficiently to protect us from infection. But clearly something doesn't quite work properly because we do get infected with SARS-CoV-2 and it does cause disease. And so we've been asking, why is it that our immune systems don't deal completely with this virus on their own? In my lab, we've been very interested in a part of the immune system called the natural killer cells, or NK cells. What happens here is that a cell becomes infected and it puts proteins up on its cell surface that basically say, help, something's gone wrong. And the NK cell will come along, recognize that signal, kill the cell and get rid of the infected cell. And so we tracked a number of these sort of danger or stress signals. You can see some of them listed here, ULBP2 nectin 1, for example. And if we infect these cells, what we should see is these proteins going up on the cell surface in order to signal to the NK cells to come along and attack. What we actually see is these proteins going down. And what we were able to work out is that the SARS-CoV-2 virus has a way of getting rid of these proteins. And if it gets rid of them, the NK cell can't tell that it needs to kill off the infection. And you can see that in the bottom here. These are the cells before the infection, and we're looking at how well the NK cells kill them off. These are the cells after the infection. And you can see the NK cells can't cope as well because the virus has attacked all the signals that it needs to see. And so clearly, if we can stop the virus doing this, then our immune systems are going to be a lot better at controlling our infection and our disease. Here's another study from a group here in Cardiff. They were interested in tracking the way the virus was evolving genetically as it spread around the UK. And so they identified a particular virus variant, which started off in London and then it spread all around the UK. And they wanted to know, why is this virus variant being so successful? Why is it spreading so well around the UK? And they were able to show that the particular mutation that this virus had was in a part of the virus that is recognized by the T cell part of the immune system. So T cells recognize a little bit of the virus which ends up on the cell surface. And if the virus can change that part of itself, then the T cells find it harder to see that the cell is infected. And so you can see here in this graph at the bottom, we're looking at how well the T cells are attacking the virus. In the this color here is the original virus that started off the pandemic, and in red is the virus that's had this little mutation in the UK. And you can see this red virus, the T cells simply can't tell that this virus is there. And so that's really important because it helps us work out how is the virus evolving and how is it becoming better at evading our immune systems, and therefore how do we need to change our vaccines to keep up with the virus and to make sure that they stay effective. More recently, we've become very, very interested in long COVID. This was incredibly unexpected. You know, that the COVID-19 pandemic as a whole was potentially unexpected, but long COVID even more so. This is an incredibly debilitating condition. We have probably over 2 million cases or 2 million people with long COVID in the UK right now. Many of these people are unable to work for 6 months, 12 months, 2 years in some cases. This clearly has a massive impact on the economy and is a massive burden on the NHS to care for these people. And still today, 3 years into the pandemic, we don't really know much about this condition. We don't have good ideas, good ways of diagnosing it. We don't have really any treatments. We urgently need much better information on this condition so we can care for those people. And so here in Cardiff, we're working with our clinical colleagues that run the long COVID clinics to really take a very holistic approach to understanding those patients, try and work out what's going on. So we're doing everything from talking to the patients, understanding their disease, their conditions, their symptoms, to understand what it's like for them, as well as doing a whole suite of biological analyses, whether that's looking at their organs, looking at the virus they were infected with, looking at their immune cells, looking at their antibodies, looking at the soluble factors in their blood that control how their immune system functions, all with the aim of working out what's going on and what markers will tell us who is more at risk from long COVID, and hopefully, ultimately, how we can potentially treat this very debilitating condition.
So clearly we've been really busy during the current pandemic, but history tells us that more pandemics in the future are very likely. As Colin mentioned, in 1918, the world was hit by the Spanish flu, which killed between 20 and 40 million people worldwide. And this picture here depicts the terrible situation that we were faced with then. Now, the SARS-CoV-2, the virus that's caused COVID-19, is actually the third coronavirus to jump from animals to humans in the last 20 years. In 2003, it was the original SARS. In 2013, it was MERS that came from camels. So chances are at some point, other coronaviruses may come into humans. In 2009, it was swine flu that we were worried about and how much that was going to spread within the human population. Of course, in the news now, avian flu, H5N1, is a major concern given its high frequency within the bird population globally. In 2013 to 2016, it was Ebola virus that we were worried about. In 2016, also Zika virus was a big problem globally. And of course, then came COVID-19 in 2019, and then really globally in 2020. So unfortunately, it is inevitable that other pandemics will occur in the future. And it's also very likely that they occur at higher frequency. And this is because viruses that can cause pandemics are part of the microbe community that are just part of wildlife diversity in the wild. And the problem is human behavior is making it more likely that we're going to see more of these viruses. And that's because we're degrading land. There's loss of habitat of the wild animal species that carry a lot of these viruses. So as a consequence, humans, animals are coming closer together. Also, there's more chances of spillover from wild animals into domestic animals, which of course we interact and live closely together with. So then there's more chances of viruses jumping from these populations into humans. The other issue we have is mosquitoes and global warming, because as the world warms, mosquito populations that can carry certain viruses actually spread more around the globe. So, for example, you would have seen in the news recently concerns about dengue virus, which is a virus that's transmitted by mosquitoes, and how the mosquito species that carry dengue virus are spreading further into Europe, and at some point maybe in the UK, and transmitting this virus here in Wales. So clearly we've been busy, but there is a lot we can do to prepare for future pandemics. And I think this quote on the right-hand side really sort of captures how a lot of virologists feel. Yes, we've been really busy today. We've got a lot to do today, but we've also got a lot to do tomorrow. And we know what we can do to help protect us from future pandemics. And I think, you know, a real take-home message is that this is not all doom and gloom. I think one of the things in COVID-19 that's really come out of this terrible time is really game-changing technologies that will help us in the future. And this is particularly the case in vaccines, where really there's massive advances in vaccine technology, and as a result have been rolled out globally to great effect. And one of the things we're doing in Cardiff now to help prepare for future pandemics is to make these vaccines even better. So you'll remember the AstraZeneca COVID vaccine, there were links with very, very rare harmful clotting events in some people. And what we've been doing is working with other people globally is to understand how that occurs and identify where in the vaccine was triggering in some very rare events these clotting processes. And by identifying where the problem was in these vaccines, we can make new vaccines and design them so this does not occur in the future.
As well as looking at improving the safety of vaccines, we're also really interested in how we can make them more effective. In my lab, we're very interested in the way that antibodies control infection. You'll all be familiar, I'm sure, with the idea of neutralizing antibodies from the various the ways that's been used in the media quite a lot. These are antibodies that stick to the virus and stop it infecting your cells. But antibodies actually have two different roles. One is to stick to the virus and get rid of it. The other is to stick to your infected cells and get rid of them. And if you are infected with the coronavirus, then your body does both of these things very well. They are a key way that it gets rid of the virus. And you can see that in the graph on the right-hand side here. We're measuring how good this activity here is. And in the blue line is the blood from someone who's been infected. And they're doing this really, really well and very effectively. But surprisingly, when we test people who've been vaccinated in the red line here, you can see they certainly do this activity very well. This is what they were designed to do. But unfortunately, they don't do this activity very well. And so clearly our current vaccines have room for improvement. And so we're really interested in understanding why is it that our infection does both of these things, but vaccination only does one of them, and how can we add this activity to our vaccines to make them better in the future. Another area of really intense interest internationally as well as in Cardiff right now is that our current vaccines are great at inducing an immune response, but it's not enough to induce an immune response. That immune response has to be in the right place. And so you'll all know, you'll had your COVID vaccines, you'll all know that they are given into the muscle of your arm normally. And that's a very effective way of inducing a really good immune response. But for a virus like SARS-CoV-2, like the coronavirus that causes COVID-19, that's a virus that infects your lungs, your nose, and your mouth. And so you don't need an immune response in your left foot or your right hand. You need the immune response in your lungs to control the virus where it is. And actually, if we give you the vaccine in the arm, it does do that to some extent, but nowhere near as well as we want. If you want to generate a good immune response in the respiratory area, the best way to do that is to give the vaccine up the nose. The problem is that all of the the vast majority of the vaccine platforms that we have, which work really well if you give them in the arm, don't work as well if you put them up the nose. And so a major area of research now is to understand how we build those next generation vaccines so we can give them up the nose and we can get the immune response in the right place at the right time in the right way.
So as well as designing better vaccines, we're also spending a lot of time and effort in trying to predict what viruses we need to make vaccines against. And here in Cardiff, we're focusing a lot actually on the Amazon rainforest, because as you know, deforestation has occurred for a number of years. And of course, this habitat loss I told you about earlier is very dramatic in certain regions of the Amazon forest. So it's bringing humans and these viruses closer together. And this is really depicted quite dramatically, I feel, by this picture here, which is in Manaus, which is the capital of the Amazonas state in Brazil. And you can see how humankind is encroaching into the forest, house by house, street by street, like tentacles into the forest. And of course, these areas are the real hotbeds of risk of transmission of virus from wild animals, either directly or via mosquitoes, into humans. And so we're working with colleagues in Brazil to study this in greater detail. So we are developing large programs of work with colleagues at Unicamp, which is a university in Sao Paulo state that's a strategic partner of Cardiff University, and also with colleagues at the Fiocruz Amazonia and Manaus. And these are pictures recently where we visited the scientific facilities, and also in some of these places here, we're really on the front line, but within the Amazon rainforest and helping care for people that are presenting with various diseases in these regions. And really what we're doing with colleagues in Brazil is to understand, certainly, firstly, the viruses that already seem to be species jumping from the Amazon into humans, and understand how we can protect people from these viruses, but also trying to set up large surveillance programs to try and see really early on which other viruses are trying to do this, trying to jump from the animal populations within the Amazons into humans. The other thing we're doing is studying harmful immune responses to viruses. So something we learned in COVID-19 really is that although we need the immune response to help protect people from SARS-CoV-2 virus, actually too much of an immune response is a bad thing, and it causes disease in a process called inflammation. And something that happened during COVID-19 was that classic anti-inflammatory drugs that are used to treat various diseases underpinned and caused by inflammation were actually used very successfully in severe COVID-19 patients. So the classic example, which you would have heard about in the news, is dexamethasone, which is just a very cheap anti-inflammatory steroidal drug that was used to great effect in severe COVID-19 patients. And actually the first COVID patient to receive this drug was here in Cardiff. And what the scientific community here is doing is really doing a lot of basic research to understand how viruses cause inflammation, understanding how we can stop those processes with a view to identify new pandemic-ready therapies to treat people that are suffering from severe disease caused by inflammation in future pandemics.
And then finally, the other thing that's happened to a huge extent during the pandemic is the setting up of large structural projects within Cardiff and across the whole of Wales to track and understand the spread of this virus. And so, for example, if you are infected with the virus, that virus comes out in your waste, that ultimately ends up in a sewage treatment plant. And so we can take the water in the sewage treatment plant and we can test it for viruses. And that's a really, really good early warning system to tell you how much virus is around, and if you sequence it, it can tell you exactly which virus is around. And so, for example, if you start seeing an uptick in the amount of virus genome in your wastewater, that's a very good sign that in a few weeks' time you might see increases in case numbers in hospitals. And so that's has been ongoing over the last couple of years within Wales. And now we're beginning to ask, we've been doing that for COVID, how can we start broadening this program out and look for other viruses and other diseases, particularly looking at things like flu. If there is a case of flu within Wales, we want to know. We want to know about it early and quickly so we can do something about it. And this is a really good way of doing this. Ian also mentioned within Cardiff, we had our own asymptomatic testing pipeline. We were obviously working in the lab during the COVID pandemic, observing all the rules, of course, but there was still a chance that someone could be infected with COVID. And so by regularly testing our staff, we could make sure that if there was a case, we knew early, we knew quickly that person could isolate before it spread within the lab. And so now we have this huge expertise, all these robotics and systems ready to do large scale testing within the university. And so as with the wastewater, we're beginning to ask, how can we repurpose this pipeline now to make a difference over the next few years? And so to conclude, hopefully we've given you a very brief flavour of the kind of things that we as virologists and immunologists within Cardiff University got involved in the pandemic and tried to help out. Obviously doing our research on our own research and with colleagues around the UK and internationally, our science was clearly very important, helping out Welsh Government to ensure that they were able to develop evidence-led policy. Working with local media stations to ensure that the public were kept informed and had a trusted source of accurate information as to what was going on. And really now it's about how do we build on the legacy of the pandemic. It was obviously a terrible time for many, many people. But if we look at it scientifically, we've learned a huge amount. All scientists internationally came together to really advance our understanding of virus disease and vaccinology. And of course, the science we've learned here is relevant to all viral diseases. And so really now, as we look forward, it's how do we take what we've learned and build on it to use our science more readily for the betterment of human health internationally. I think that's, we'll leave it there. Thank you very much. Hopefully that was interesting.
That was great. Thanks very much indeed, both. So we've now got about 20 minutes, well, just under 20 minutes actually, for questions. We'll just take the shared screen down there, be able to see us. And we will finish punctually at 6. There's a couple of questions. There are some questions in the chat, which is in the Q&A. That's really great. Please keep them coming in. I'm going to start with a one or two pre-submitted questions, then I'll come on to the ones that you've been putting. So keep putting those questions. And if we don't get round to all the questions that have been asked, we will answer them as well. Ian and Rich will answer them for us. So you'll get an email subsequent to this, but we'll try and get around to as many as we can. Any that we can't, you will nevertheless still get an answer to. So please do go ahead. Okay, so the first one is one of real interest to everybody, I think. When do you expect the next generation COVID vaccines to become available that will limit or even stop transmission of the virus? You know, this thing that sadly didn't happen this time around.
Yeah, so I think this really goes to the heart of one of the slides I put up, saying about getting the right immune response in the right place. I think if we want to stop the virus transmitting, we have to have the immune response in the nose, in the mouth, in the lungs. That's really, really critical. And I think sadly we are going to be a few years away from that at the moment. It's quite a complicated situation. Our scientific understanding of how we make those vaccines that raise the right type of immune response in your mouth, nose, and lung is not where it needs to be yet. That's absolutely something absolutely critical we have to work on for the next few years. It's also really complicated at the moment testing these vaccines because, of course, when we started off the vaccine program, no one had immunity. to the virus, so it was a clean slate. Now everyone has very different types of immunity. Some people have been just vaccinated, some have been vaccinated and infected once, some have been infected by Alpha and then Delta and then Omicron and then Omicron 2 and Omicron 3 and Omicron 4. And so running the clinical trials to understand how effective your vaccine is really, really difficult. So it's something everyone is really working very hard on, but I think we are a few years away from that. That's my personal opinion. I don't know.
Yeah, I agree with that.
Okay, great, thanks. And another one which I think some people will be very interested in, since COVID obviously is still circulating in the UK, I mean, it's endemic now, isn't it? How dangerous is it now for clinically vulnerable and extremely vulnerable, well, clinically vulnerable people?
So, I mean, we've done a lot of work in Cardiff, for example, in kidney transplant patients, and there's variation in terms of how different people within those patient cohorts respond to the vaccine. And certainly, so for example, in hemodialysis patients, it was quite a problem after 3 vaccines that they still weren't inducing a good immune response. But actually, 4th one suddenly made a big difference. So actually, these continual boosting for these individuals does help. There is still some intervariation between different people within those cohorts. So actually having blood tests and understanding how each individual in a personalized medicine way responds to the vaccine will really help guide people's decisions and how they behave, particularly at times when COVID the virus is quite common in the population.
I think it also raised the issue that we have the vaccines, and the vaccines are great, but they're not perfect for everyone, and so we need to keep on the development of alternatives to the vaccines as well. There are antivirals and immunotherapies and monoclonal antibodies, all these other approaches which are going on in the background. It's really, really important we have those, because those are the kind of thing that will work very effectively in people who don't respond to the vaccine. So the vaccine is great for many of us. Currently, it's not enough for all patient groups, and we need these alternatives too.
So it's personalized medicine, isn't it? It's the specific person, especially for vulnerable people. That's great, thanks. I'll take one from the Q&A now. And this is interesting. What's the difference between long COVID and ME? Other similar sort of long-lasting viral things.
Oh God, yeah. So that's a really good question and a really difficult one. That there are a huge number of similarities between ME and long COVID, and a lot of people have drawn those similarities and said, actually, what can we learn from what's been done in ME to try and drive what we do in long COVID? I think really the depressing thing is that we've known about ME for years and years and years, and we don't know what it is yet, and so. It's a very difficult disease to understand. The big problem with both ME, I think, and long COVID is that it's not the same for everyone, and the cause won't be the same in everyone, and the solution won't be the same in everyone. And so trying to pick apart these diseases where it's different in everyone is very, very difficult. And people have been working on ME for years now and haven't made the progress that we would have wanted. So, it's a very good question. It's a very difficult one.
I'd say the optimist in me, one difference is, we know that SARS-CoV-2 causes long COVID, and with ME, the actual cause of that is still up for debate. So that puts us one step further ahead in the process in terms of certainly understanding how the immune response behaves. At least we sort of know what we're working with. But the challenge is the heterogeneity, how different people present long COVID in different ways, and it's not going to be a one-size-fits-all approach to treating them for sure.
Okay, thanks. Somebody's asked whether references are going to be made available. I'm sure that's okay, isn't it? So I think we can just say, we'll absolutely do that, because there are people who are interested in the research. There's just going back to the actual Q&A. So many questions here. I need to sort of choose one. Yes, there's one here. How was from John Ridley? How were all the UK research efforts coordinated to ensure there was no duplication of effort? Probably there was some duplication, but coordination of it.
It was really interesting, actually. I think the way science was done during the pandemic was very different to how we usually do science. And it was actually, as someone who's involved in that effort, I really enjoyed the way we did science, because very early on in the pandemic, the funders put calls out saying, we'll fund research if you come to us with your ideas, and everyone piled in, and they looked at all the applications and they said, well, you're all doing the same thing, we can't fund the same thing more than once, it would be crazy to do that. And they basically said to us, you arrange yourselves into groups and you work together and you don't duplicate effort. And so we had these large consortia. Ian mentioned the UK Coronavirus Immunology Consortium that we were part of, and they were really, really good fun. Actually, I really enjoyed doing science in that way, because you get exposed to all these other ideas, you can bounce ideas of other people, you get exposed to ideas and techniques you wouldn't have come up with on your own. And so that was really exciting. I really enjoyed doing science in that way, and I think it's going to be really interesting to see if we can maintain that way of science going forwards, rather than just being people working in their little silos.
And I think a silver lining of the pandemic is that is happening to some degree. So there's more emphasis from funding agencies about generating research networks and bringing people together and getting people talking in the same way that we did during the pandemic.
Somebody's asked an interesting question about what the next pandemic might be, whether avian flu is going to be the next pandemic, and obviously H5N1, for example. Obviously, we can't really know that, but I guess what would be very interesting to hear from you is there a danger that, just as with COVID, we kind of reacted as though it was going to be flu and it wasn't? Is there a danger that we react to the next pandemic as though it's COVID and it isn't? Or have we learned enough generic stuff to be able to cope better?
Well, I think there's certain generics. I think we can make certain predictions of the type of virus that's going to cause the next pandemic, particularly the type of genome it has. So then you can sort of narrow down to what's going to be certain families of viruses. And that's the kind of work we're doing in Brazil, for example, is focusing on pandemic type viruses. So there's certain ways that they will be relatively similar, but then there's also many differences with how they transmit, etc. But that's why I think it's important to look at lots of different types of viruses we think may be likely, and get some idea of the basic biology with the types of viruses from a particular family we know about, say types of flus, types of coronaviruses, types of other viruses, to get some broad lessons from those. There will be differences though in terms of transmission, because that depends entirely on that virus, how rapidly it's shed from that individual. I mean, one of the problems with SARS-CoV-2 was that people were quite infectious before they actually presented with symptoms. And that was a big issue with this particular pandemic that may be not the case with others. It certainly wasn't the case with the first SARS-CoV virus. So even a similar coronavirus was behaving quite differently in humans, which affected how much of a problem, how much it spread within the human population globally.
I think there's also lessons to be learned beyond the science as well. I think if we look globally at the response to the pandemic, places that were either had or close to the SARS-1 outbreak or the MERS outbreak responded much more quickly and very differently to those of us that hadn't really experienced the pandemic before. And actually their response was faster and better than ours, because they'd had that previous experience of being so close to having something very dangerous spreading in their countries. And so I think that again potentially is a bit of a silver lining, in that now we've had this experience, we might be more willing to act more quickly next time.
There's a couple of questions that actually go in that direction. One about sort of the non-clinical intervention, the behavioural aspects and what we've learned about that, and one about mistrust in official scientific or government communications. We all know about the conspiracy theories, the anti-vax movement and so on. What have we learned there? Because we can have all the wonderful kind of medicines we like if we can't get people to take them. So yeah, what are your thoughts on that?
I think that's a very well-made point. I think, we're basic scientists, we work in the science world. Prior to the pandemic, if you'd asked me how we were going to solve the pandemic, I'd have said it's science all the way. But actually, for the first year of the pandemic, we were saved by the behavioural scientists. We were the non-pharmaceutical interventions were what saved us for the first year. And we're not behavioural scientists, but we certainly worked with behavioural scientists in our advisory groups to Welsh Government and that sort of thing. And I think the things that they tended to make, the points they tended to make were things like, whether a population trusts the government has a massive impact on whether they are willing to follow those rules. And the way the government does its messaging is so important in whether people are carried along for the ride and we're all in it together, we've got to help each other out, or if it becomes you're being forced to do this. And so that's not our field, but there are many researchers in Cardiff and around the UK who do work in that field, and it's a really, really important topic that's being actively investigated right now.
I think just to add to that personal note, so myself and Rich were involved in the International Intelligence Group in Welsh Government, where we were looking outwardly at what other countries were doing, how would they do things the same or different, and what didn't. And so within that, there's a lot of statistics globally about how well different countries align to what the government were telling them. And I think there's a lot to be learned looking at, as things are somewhat calmer, to see what other countries did very well, maybe better than us, and how we can sort of learn from that for future pandemics.
Yeah, fascinating. We've got more questions coming in. Just sort of adjacent to this, perhaps. Why has it been so difficult to identify the source of the COVID virus? I think I can guess the answer to this, but what's your view on that?
So another a very controversial topic at the moment. I think in part it is, well, by definition, our ability to assess what happened in those early days was limited, because as soon as they realized something was going on, they went in there and they closed the markets down, and that got rid of all the evidence we could potentially have found there. Along with the fact, I mean, I don't want to get political, but the Chinese government traditionally is fairly secretive about many of these things, which immediately people start seeing things that aren't there, or inventing things where they can't see evidence. I think certainly the vast majority of scientists, certainly that I know, would favour the hypothesis that it jumped within that wet animal market from animals to humans. That's certainly, in my opinion, that's where the evidence lies currently. But I suspect we will never know for certain.
Okay, we've got just a couple of minutes to go. Really good, interesting question here. Is there a key thing or several things which governments would do differently if another COVID-type pandemic were to arise? And are there any of these things which came directly from Cardiff? So can we identify anything that we've really contributed that would change the response next time?
Well, certainly, I think we will lock down earlier. I don't think that we can say that's the Cardiff-led thing. I think certainly one thing that we're involved in, I was personally involved in, is vaccine strategy and identifying how spread within care homes and we were involved in within hospitals is really important, and getting these people vaccinated earlier. That's something we've personally done, and Cardiff have personally been involved in.
Yeah, I think much of the ongoing research really now is about developing those better vaccines, learning now how do we, the COVID vaccine was let's pick the most likely thing that we think will do something and get it as quickly as possible into people. What we need to do now, what we are doing now, is saying, well, actually, let's be clever about this. Let's not just pick the one thing, let's pick lots of things, and let's try and work out what properties we want our ideal vaccine to have, so that when, if there is another outbreak, and of course, we're talking about another outbreak, actually, everything we're talking about is relevant to seasonal flu as well. The scientific advances we're making here are not just for pandemics; they are for all viruses that infect us. Sorry.
Yeah, I mean, just one thing outside our sphere of expertise is also, governments are very worried about next time. How do they get enough face masks? How do they get enough equipment? How do they get enough things? And there's certainly a lot of work in the background going in there, and also, working with mathematical modelers elsewhere in Cardiff. How do you best operate a hospital during COVID times, because there's a lot of data out there. So data scientists are looking at what's happened or what happened before, and how can we improve that in the future. So there's a lot of different things from different angles that people are doing that we can learn from for next time, for sure.
Yeah. And it's worth saying that, Welsh Government are very open to this, and we're in active discussions at the moment with all the universities around Wales and with Welsh Government to try and understand what do we need to put in place now to make sure that we can respond better in the future. So those discussions are ongoing and are happening.
I must say, from a personal point of view, what I learned managing the whole thing from a Cardiff University point of view was that we did a lot of really fascinating and useful mathematical modelling of how we would, get the students to behave and so on, but they didn't actually behave that way. So I think bringing together the modelling with what we learned about the way people genuinely behave will be really useful. But we've run out of time. That was absolutely fascinating. We've got loads of questions we didn't get to, but we will get back to you all on that. Massive thank you to Ian and Rich. That was really interesting, and I know the audience found it interesting as well. And as I say, we will follow up on the questions. So you should receive an email in the next few days with a copy of the recording and links to register for future events. The next one in the series takes place on Thursday, the 8th of June. And that's also a hot topic, which is cybersecurity and artificial intelligence. We're all now, dealing with the prospects and consequences of AI, and that's what we're going to have a session on: how to keep computer systems safe from the threat of cyber attacks, and how to manage the opportunities and potential threats of artificial intelligence. Now, if anyone watching would like to get involved with Cardiff University, volunteering time to help our students or helping us to fundraise for our research, please do follow the link in the comments box. It's just gone up there, I can see. That's it for now. Thank you all very much for attending. Thank you and goodbye. Diolch a hwyl fawr.